Hello and welcome everybody to my channel Omni Bioentrance, and today we will be discussing ICMR BRET question paper. This was conducted in the year 2024 and I am discussing the section B questions, that is for cross-disciplinary questions I will be discussing. If you are new to my channel, please do like, share and subscribe. If you want still previous year question papers of ICMR being discussed, you can go to my channel Omni Bioentrance. In the channel playlist you see for ICMR and there you will find. So coming to the question. The question is with regard to match the following. Match the list 1 with list 2. So tag DNA polymerase, you know it is used in PCR for amplifying DNA. So A becomes 3 used to amplify DNA. A3 you can see in option 2 and option 4. So 1 and 3 we need not take into consideration. Then MMLV reverse transcriptase. Reverse transcriptase is for synthesizing DNA from RNA. So it is used to make cDNA. So B1. So 4 is B1. Then RNA polymerase 2. So option 2 is also wrong. Okay. So 4 will be the answer. Okay, we will see. Then RNA polymerase 2, it is for mRNA synthesis in eukaryotes. So C will be 4, that is transcription particularly of mRNA, lncRNA. So C 4. Then T for polynucleotide kinase, it is for phosphorylate oligos or DNA. Kinase is usually for phosphorylation reaction. So here it is used to phosphorylate oligos or DNA. So D2. So answer will be option 4. Then next one, select the correct primer pair of forward and reverse primer sequence that amplify full length sequence given below. So this is your DNA which has to be amplified. This is the flanking sequence. Okay, so from the flanking sequence you have to design your primer. You have to see which is your forward primer and which is your reverse primer. They have given both in the 5' 3' direction. Okay, we will see. Now this kind of question, that is for asking what is the sequence of the forward primer, what is the sequence of reverse primer, these are regularly asked in CSIR exam also. So here is the sequence. I have rewritten it over here. This is 1 to 100 to 1. And this is the flanking sequence. Now in small letter I have written the primer sequence, and this is the capital letter, it is the sequence what they have given in the question. Okay. If you see here, this will be your reverse primer. Okay, this one. So the strand 5' to 3' will give the reverse primer, and this strand, that is the complementary to the 5' which I have written here. I have made the complementary sequence and written. They have not given the complementary sequence in the question, but I have written that for you to understand. So this is the 3' to 5' sequence, and that strand will give you the forward primer. So first we will see the forward primer. So complementary to this 3', 5' strand, we will have the forward primer. So it will be TAC, TAT, CGA, GTA, TAG. If you see the sequence, you can see that this is the same sequence as given in this 5' to 3' strand. So if you want the forward primer, then you have to see you can take the sequence of the 5', 3' strand as your forward primer sequence. So that itself will give you the forward primer sequence. So TAC, you can see in option 2 is there. Okay, so that will be the answer. They have not given TAC in any of the other sequences as your forward primer. So first one gone. Third one it is ATG, so that also gone, and fourth one it is a TAG, so that also gone. So the answer will be second. Now for your reverse primer. So this sequence, this way, okay, in this direction, that is 3' to 5', this direction will give you the reverse primer sequence, and they have given it in the 5' to 3' direction. So GTA, GTT, GC, see here if you see GTA, GTA, then ACG, okay, GTA, GTA, okay, ACG is here, then TAC, okay, TAC, yeah, TAC, and then GTA, CG. So that is your reverse primer. So that's it with regard to the primer sequence. Now coming to the next question. So dash variant of BLAST compares a DNA query sequence to the protein database. So here if you see this table, you can see here in this column they have given the query sequence, and here is the query sequence is compared to which sequence in the database they have given, and the program required for this comparison, that is the BLAST program that is required for this comparison, is given in this column. So if your sequence is nucleotides, that is just your DNA sequences, and you want to compare with the nucleotide sequence in the database, you go for the BLAST-N program. Now if your query sequence is protein, and your query sequence means the sequence that you are submitting to the database, your sequence. Suppose you have done some experiment in the laboratory and you got a new sequence, and you want to see from the database to which protein this particular sequence is matching, you want to find out. So you go for first you do the nucleotide to nucleotide sequence comparison using the BLAST-N program. Then you do convert the nucleotide sequence to protein sequence, and then you do the protein to protein comparison using the BLAST-P sequence. Okay, I mean BLAST-P program. Okay, now you have the nucleotide translated into protein, that is your query sequence, and you want to compare it with the protein database, you go for the BLAST-X. Now your query sequence is your protein, and you want to the database should be the nucleotide translated into protein sequence, then you have to go for the T-BLAST-N. And if your query sequence is nucleotide translated into protein and you have to match it with the nucleotide translated protein in the database, then you have to go for the T-BLAST-X. Now here what they have asked is DNA is the query sequence, that is your nucleotide is the query sequence, and you have to match it with the protein database. So nucleotide protein will be this one, BLAST-X. Nucleotide translated into protein, match with the protein database will be the BLAST-X. It cannot be T-BLAST-N because T-BLAST-N the query sequence is protein, but in BLAST-X the DNA query sequence is used, and in the question also they have asked DNA as the query sequence, that is your nucleotide should be the query sequence. So the answer here is BLAST-X. Then next one, which one of the following is not a primary database? Now what are these primary and secondary database when you are learning bioinformatics? So primary database, you know, it is just the raw data, whatever you get after conducting an experiment in the laboratory. That is, you get a new sequence from a new organism, and you are submitting it into GenBank of NCBI. It is nothing but your primary data. Okay, that becomes your primary database. It can be either nucleotide sequence or protein sequence that you are submitting. So these data are directly submitted by researchers or by various labs conducting various experiments. That is your primary data. And primary database examples include GenBank. It is for DNA sequence submission. Then a protein data bank or the PDB is there for three-dimensional protein structure, and EMBL and DDBJ are also other primary databases which are for nucleotide sequence submission. Okay, that is primary database. Now what is a secondary database? So the primary data, whatever you have submitted, that data will be analyzed, interpreted, annotated by these computer programs using various algorithms, and expert review also will be conducted, and finally they become the secondary database. And these secondary database, they provide the processed and curated information to various researchers, and it also gives information regarding the various protein motifs, then protein domains as well as protein families also they give information. So that is your secondary database. Examples include Uniport, Swissport, then PFAM and ProCite. These are mainly for the protein families and domains. So they have asked which is not a primary database, so that makes Swissport, which is a secondary database. So EMBL, DDBJ and GenBank, they are primary database. Then next one, the computational approaches to predict the three-dimensional structure of protein. They have asked for computational approaches. That means this X-ray crystallography, NMR, they are your experimental approaches to determine the three-dimensional structure of proteins. Computational means you are using various bioinformatic tools to determine the structure of proteins. Now first one, it will be your homology modeling. And homology modeling, it is based on the principle that a protein with the similar sequence, that is if you have 2-3 proteins, they are of the similar sequence, means they will have the same structure or similar structure, not the same structure, they will have similar structure. So what you do is you submit your protein into the database, sequence into the database, and it will predict the structure of this particular protein that you have submitted based on the protein structure information which has been loaded in the database. So database is already having different protein structure information. So it will compare your sequence of the protein that you have given with various structure present in the database, and it will predict and tell, see this protein whatever you have discovered in your lab will be having a structure similar to this particular protein. So that is homology modeling. Now what is threading? Threading is also called as fold recognition, and it will predict the structural fold of unknown protein sequences. What it will do is it will fit this unknown protein sequence into the structural database, and it will select for the best fitting fold. Now these two process approaches, that is homology modeling and threading, they take information from the sequence that is already stored in the database. Now if you don't have any structural template to be compared for your protein of interest in the database, then what you will do? You will go for what is called as the ab initio approach. So in this, it will give you the sequence, structural information regarding the protein without the use of any structural templates which are found in the database. Okay, so it will predict the protein structure from just on the sequence information that you have provided. So it will analyze the protein sequence, and it will predict the structure of the protein based on its physiochemical properties of the protein, that is the physical and the chemical and the statistical properties of the protein will be taken into consideration, and it will tell you, see this is the protein folding that can be given to this sequence that you have generated in your lab, and this is how the protein structure will look like. All this information you can get from ab initio method. So that makes A, D, E as the answer, and your option is 3. Then next one, before coming to the question, we will see how iron is taken up by the transferrin receptor. Now this is your cell membrane. See this portion. This is your cell membrane. These blue ones, that is DMT1, that is divalent metal transporter 1. These dots, they are your Fe2 plus, and this is your apotransferrin. It is called apotransferrin because it is not bound to any iron, and this is your transferrin receptor 1. Now once the iron binds to this transferrin, it will bind to the transferrin receptor. This apotransferrin, it will bind to the TFR1 receptor, and this transferrin bound receptor will be endocytosed endocytosed in clathrin coated pits. Okay. Now these clathrin coated vesicle, they will lose the clathrin coat. How? That because there will be reduction in the pH of this in the interior of this particular endosome because of the presence of proton pumps. So these proton pumps, they will inject H positive ions into this endosome, and when there is reduction in the pH, the clathrin coat will be lost. And again there will be influx of the H plus ions into the endosome, and once the pH decreases to about 5.5, it will result in the dissociation of the iron from the transferrin. Okay. Now what will happen? The iron will be transported out of the endosome via the DMT1, that is your divalent metal transporter 1, and this iron which is released, it will be incorporated into the heme, or it will be taken for storage into ferritin, or it can be used for some other metabolism of the cell. Then after this receptor and the transferrin, so now the transferrin, it is not bound with the iron, so it is apotransferrin, and this apotransferrin which is bound to the receptor because the pH is reduced inside this endosome, and the transferrin has got a high affinity for its receptor at a reduced pH, and this whole thing, it will be taken to the cell membrane, and because the cell membrane, it is having a neutral pH there, this transferrin and this transferrin receptor and apotransferrin will dissociate from each other, and they will be available for the next cycle. Okay, this is what is happening in iron uptake by this transferrin receptor. Now come to the question. What happened to transferrin when iron is bound to it and it interacts with its receptor? So we already saw the whole process. So is it degraded inside the lysosome? No, it is taken up by the endosome, and upon reduction of the pH in the endosome because of the presence of proton pumps, iron is dissociated from the transferrin. Iron is dissociated from the transferrin, and the iron which is dissociated will be transported out of the endosome into the cytosol. So it is not degraded inside the lysosome. So that makes option 1 gone. Undergoes a conformational change allowing binding to its receptor. That is correct. Now this transferrin, when it is bound to iron, there will be a conformational change in the transferrin, and so it can bind to its receptor more tightly. Okay, so that can be correct. Then it is converted to apotransferrin. No, Once transferrin is bound to iron, it is not apotransferrin. If transferrin is not bound to iron, then it is apotransferrin. Okay, so when transferrin, when bound to iron and it interacts with its receptor, it is not apotransferrin, it is holotransferrin. So 3 is wrong. Permanently internalized and degraded, no. Iron is released into the cell, into the cytosol. So it is not degraded. It is neither permanently internalized. The transferrin bound to its receptor is again recycled back to the cell membrane. So it is not permanently internalized and degraded. So 4 is also not correct. So the answer is option 2. Now before coming to the next question, we will see what are the various non-viral gene delivery techniques or methods. So there are 2 types, one is physical and the other is chemical. So chemical means it will use various substances to carry genetic material and deliver it into the cell. So one of the method is by liposomes or by lipid nanoparticles. So these are liposomes and lipid nanoparticles. They are vesicles with a lipid bilayer and it can encapsulate small RNA and DNA molecules. And this encapsulation is for protecting the genetic material from degradation, and it will aid in cell entry of this genetic material. So that is the method used by liposomes. Then there is another method that is polymer based. So these polymers, they can be cationic polymers or biodegradable polymers. And these polymers, they form a complex with the DNA, and this is called as polyplexes, and they are taken up by the cell. So that is how the DNA is delivered into the cell using polymer based method. So cationic polymer example is polyethyleneamine, PEI. And biodegradable polymer is your polybeta amino ester. Okay, remember these examples, they may ask for your next exam. Then there is another chemical method that is by the use of peptides. Peptides, you know, they are short chain amino acids of 2 to 50 amino acid length, and they will interact with the genetic material, that is your DNA or RNA, which has to be delivered into the cell, and they form what is called as peptiplexes. Polymer based, it was polyplexes. Peptide based, it is peptiplexes. And they will deliver the gene through conjugation or through electrostatic forces, interaction, etc. So that is how peptides deliver gene into the cell. Example of this peptide include peptide nucleic acid or PNA conjugates. Then there is something called as dendrimers. Dendrimers, these are highly branched macromolecules. They are spherical in shape and on their surface they have positively charged. So your DNA and RNA, which is negatively charged, can bind to the surface of these dendrimers, and this whole complex is being taken up by the cell through endocytosis. Okay, that is how dendrimers deliver the gene into a cell. Now coming to the various physical methods of gene delivery into a cell. So these methods, that is physical methods, they use various physical forces to overcome the cell membrane barrier and enable the entry of the gene of interest into the cell. So first method is by electroporation, that is it will use brief high voltage electric pulses, and it will create temporary pores on the cell membrane, and through these pores your genetic material can enter. Then there is another technique called as the gene gun method. So through gene gun, the genetic material will be coated onto a microscopic heavy metal, that is it can be your gold particle, and then it will be shot into the target cell using high pressure gas. Okay, high pressure gas will be used for shooting. That is called as gene gun method. Then there is sonoporation. Sonoporation means it will use ultrasound waves to create transient pores in the cell membrane, and through these pores your gene of interest can enter. Then there is hydrodynamic injection. So in this method what you use is you use large volume of solution containing your DNA or RNA or any genetic material which has to be injected into the cell, and this is rapidly injected into the blood vessel. Okay, rapidly injected into the blood vessel, and this can result in efficient uptake of the genetic material. Then there is what is called as micro injection, that is injecting genetic material into the nucleus through your cell's nuclear envelope. That is called as micro injection. And there is magnetofection, that is magnetic nanoparticles. They will carry the gene, and they will be this magnetic nanoparticles, they will be directed to the target cell by the application of an external magnetic field, and this will enhance a cellular uptake. So that is the two methods of non-viral gene delivery, that is physical and chemical method. Viral gene delivery is through adenoviruses. Okay, usually adenoviral vectors are used. So we will see the question now. So which non-viral gene delivery method utilize physical force to introduce genetic material into cell? Liposome, it is chemical mediated. Poly-L-lysine, I told you it is polymer based, which is again a chemical method. Adenovirus, it is a viral gene delivery method, not a non-viral gene delivery method. And electroporation, it is a physical method, and it uses physical force to introduce genetic material into cell. So the answer is option 3. Now coming to what is called as RNA interference. So RNA interference, it is a natural mechanism to silence gene expression by using short interfering RNA, that is your siRNA or microRNA, and it will degrade the messenger RNA, and this is usually a means of protection of the cell from viral infection. So if the cell encounters a long double-stranded RNA from a virus, what it will do is there is an enzyme called as the dicer, and it will cut this long double-stranded RNA into small fragments around 21 to 25 nucleotides long, and these are called as the siRNA. And this siRNA is loaded into the RISC complex or the RNA induced silencing complex. And in the RISC complex you have got a protein called as the agronaut protein, and this double-stranded siRNA, it will bind to this agronaut protein, and in this RISC complex the one strand of this siRNA is separated from the second strand. So one strand and it becomes single stranded, and one strand of the siRNA will be retained in the RISC complex, and it will act as the guide strand. That is, it will find the target mRNA that has to be degraded by using the principle of complementary base pairing. And once the RISC, this siRNA finds the mRNA, this RISC then will target this mRNA to degradation with the help of the Argonaut enzyme. Okay. Now, what is the question? Which protein is responsible for cleaving double-stranded RNA into smaller fragments using RNA interference? It is nothing but your dicer protein. So, answer is option 4. Helicase, it is for unwinding the double-stranded DNA during replication. Risk, it is a complex which is involved in mRNA degradation. And slicer, it is an activity that is an inherent ability of the Argonaut family members to cleave the target mRNA molecule. Okay, so the answer is dicer. Then next one, what is the main advantage of short tandem repeats over restriction fragment length polymorphism in DNA fingerprinting? So DNA fingerprinting, it is a laboratory technique and it is used to identify individual based on unique pattern of the DNA. Okay, it is used in forensic studies, this DNA fingerprinting. So there are 3 types of markers that are used for DNA fingerprinting, and these are the first one was your RFLP, which was used, and then came the variable number of tandem repeats. Now RFLP, how it detect this DNA sequence variation is by using restriction enzymes. Once STR got popular, this VNTR and RFLP techniques have been reduced. They have reduced the usage of RFLP technique and VNTR methods. Okay. Now VNTR, it consists of longer sequences, that is about 10 to 100 base pair sequences. That is the variable number of tandem repeat sequences. There is reduction in the usage of VNTR also, and at present short tandem repeats are being used over RFLP and VNTR. So short tandem repeat are these are repetitive sequence of 2 to 6 base pair, and they are highly polymorphic, and they are distributed in your genome and widely distributed in the genome. So why do you use these short tandem repeat? Means they are easier to visualize on gel? No, they require less DNA and more resistant to degradation. Yes, that is more correct. That is why you use STR. Okay. Then, which of the following steps in PCR is responsible for separating double-stranded DNA into single strand? So what are the steps in PCR? You have got the denaturation. So this is your double-stranded DNA at 95 degrees. Heating, it will denature the hydrogen bond between the various bases are broken, and you get 2 single-stranded bands. Then once you get the single strand, you have got the annealing step where the primers bind based on their complementary base pairing, and they will start synthesizing the, or I can say they will start amplifying your gene of interest. So that is at 72 degrees centigrade, that is your extension. So these are the various steps in PCR. So what they have asked, the step which is responsible for separating double-stranded DNA into single strand, that is your denaturation step or your melting. So answer is option 3. Then match the following. Autotrophs, they are your primary producers, that is plants. So A will be 3. A3 means, oh great, only one option is giving the right answer, the others are wrong. So if you know autotroph as primary producers, you can happily go for option 3. Okay, but see the rest also. Phagotrophs. Phagotrophs are nothing but your macro consumers, and they are your herbivores, carnivores, omnivores. All these will come under macro consumers or phagotrophs. And saprotrophs means they are your decomposers. So they are your micro consumers, that is your bacteria and fungi. So C, B will be 1, C will be 2. B1, C2. Okay. Heliotroph means, helio means sun. So it is related to sun. Okay, so it will be sun loving plants. So D will be 4. Answer will be option 3. Then next one, the reductive pentose phosphate pathway is also known as, it is called as the Calvin-Benzene cycle, and it is a biochemical pathway for the fixation of carbon dioxide into organic molecule, and it uses energy from ATP and NADPH, which are produced in the light dependent reaction. So that is the reductive pentose phosphate pathway. What is photosynthetic carbon oxidation cycle? It is your C2 cycle or your photorespiration. Okay. So the answer here is option B, Calvin-Benzene cycle. B only. 3. 3 is the answer. Then next one. Cortisons used in the treatment of rheumatoid arthritis are produced from the fermentation of dash by Rhizopus migracans. So it is glycosides. It is by the fermentation of glycosides that you produce the cortisons, which are required in the treatment of rheumatoid arthritis. Then in cell cycle there are 4 stages, M, G1, S and G2. There is another term commonly used, that is G0 phase. Choose the best described statement for this G0 phase. So G0 phase, the cells are metabolically active. That is, they continue to perform the essential cellular functions, but they do not undergo any division or proliferation or differentiation during this period. That is your G0 phase. So cells are at a growing stage. No, they are not growing. There is no division. Cells are at the stage of differentiation. No differentiation. Cells have ceased to divide. Yes, that is correct. Cells are about to double. Double means they are undergoing division. There is no division in G0. So 4 is also not correct. Then next one. Based on series of events outlined in central dogma, there are enzymes that convert from one stage to another. Okay. However, one conversion is not possible yet. Identify the stage. So central dogma is DNA to RNA and then to protein. So DNA undergoes transcription to produce RNA, and RNA is translated to protein. RNA can be reverse transcribed to produce DNA, and DNA can undergo replication also. So this state, that is RNA to DNA and DNA to RNA, is reversible, whereas RNA to protein, it is not reversible. That is what they have asked. One conversion is not possible yet. What is that? Protein to RNA. DNA to RNA, yes, this one, transcription. DNA to DNA, replication is possible. RNA to DNA, reverse transcription is possible. Enzymes are also involved in all these stages, but there are no enzymes which convert protein to RNA. So the answer is option 2. Then before coming to the next question, we will see some points regarding the lac operon. You all know lac operon. You have been studying it from your school. So here is the structure of the operon. You have got the repressor gene, then promoter, operator, and the various structural gene, that is ZYA. And this ZYA, they are under the control of a single promoter. Now, in the absence of inducer, your inducer is your allolactose or your lactose. That is the substrate. So, when lactose is not present, the lac operon genes need not be synthesized for taking up of the lactose. Okay, so what will happen? The repressor protein, it will go and bind to the operator, prevent the RNA polymerase from binding to the promoter, and thereby block the synthesis of or repress the synthesis of the lac operon genes. Now in the presence of inducer, what happens is the inducer, that is your allolactose or whatever, it will bind to the repressor, changes its conformation. Repressor cannot bind to the operator. Repressor not bound to the operator means promoter site becomes free for RNA polymerase to bind. RNA polymerase will bind to the promoter. It will start synthesizing the ZYA structural genes which are for beta-galactosidase, permease and trans-acetylase. Okay. And this I told you, the ZYA genes, they are under the control of a single promoter, and they will result in the synthesis of one RNA molecule in a continuous process, and it is a polycistronic RNA that is being synthesized, and it will have the coding sequence of all the three proteins, that is beta-galactosidase, permease and trans-acetylase. So that is it with regards to lac operon. Now come to the question. So in lactose operon, the following events happen. The operon is normally in an off mode when appropriate substrate is absent. Substrate is your lactose. So when lactose is absent, structural genes need not be synthesized. So operon will be in off mode. So first statement is correct. Lactose is an inducer substrate. Yes, it will induce the synthesis of the lac operon genes. So that is correct. If lactose is added to the cell environment, it triggers events that turn the operator on. Okay, now if you add lactose to bacterial cell, lactose will enter into the cell, it will bind to the repressor, repressor will not be able to bind to operator, RNA polymerase will happily bind, structural gene synthesis take place. So it will on the lac operon, so C is also correct. Then the structural genes are transcribed in 3 different transcripts coding for all 3 different enzymes. No. It is one single transcript which is coding for all the 3 enzymes. So D is not correct. As lactose is depleted, operon is repressed as per me. It is correct, but they have not included that option in the question paper. That is E option, that is A, B, C, E is not present. So we will go for option 1 as the answer, and option 1 is what they have given in your answer key also. As per me, E is also correct. As lactose is depleted, operon will get repressed. That is correct. Okay, and wherever they have given option E, D is wrong. Again, D is wrong here. So we cannot say 3 and 4 are correct, even though C is correct here, E is correct here, A is correct here, E is correct here. So the correct answer will be A, B, C. So that's it. Thank you for watching my channel. Please do like, share and subscribe if you are new to my channel. And if you want still previous year question papers of ICMR being discussed, you can go to my channel Omni Bio Entrance. In the channel playlist, you can select for ICMR, and there you will find previous year question papers of ICMR. Thank you.